So I'll be telling you guys a bit about our Pioneer model, which is our model for designing personalized cancer vaccines based on neoantigens. So in the past 10 to 20 years, there's really been a revolution within the treatment of cancers, and that has been based on these cancer immunotherapies using checkpoint inhibitors. These checkpoint inhibitor treatments allow physicians to treat certain cancers that were previously basically untreatable with, for example, chemotherapy. And a good example of this is melanoma, where there's been a huge advance in the quality of care we can give to patients. However, when we sort of dig into the clinical trials that tested these checkpoint inhibitor treatments, we actually see that it was only around 20 to 30% of the patients in the trials that actually got a benefit from the treatment, so meaning that their tumors shrank during the treatment. So this highlights the need for improved treatments within this area. So neoantigen-based cancer vaccines are sort of the next logical step in the evolution of these cancer immunotherapies. And they are this because they arise from cancer-specific DNA mutations. And this means that neoantigens are found specifically within the tumors of patients and absent from their other cells, from their other tissues. The fact that they are found specifically in their tumors allows a neoantigen cancer vaccine to elicit strong and highly specific immune responses directly towards the tumor, and hopefully avoiding a lot of side effects that we see from current cancer immunotherapies. And we also believe that the fact that we are triggering these highly specific immune responses allow us to synergize well with the current cancer immunotherapies, which is a big advantage when you are looking to validate these in the clinic. There is, however, a challenge in developing these neoantigen cancer vaccines, and that is the fact that each and every patient has a distinct set of neoantigens within their cancer cells. So here I'm showing you a graph from a study where the authors analyzed the genomes of more than 300 tumors from melanoma cancer patients. And what you can appreciate is that the number of mutations, and thereby potential neoantigens, varied a lot from patient to patient, with some patients having less than 100 mutations and some patients having several thousand. So we can already see here that the set of neoantigens in each patient must differ. They also dug deeper and looked to sort of see where there are some mutations that maybe were found in several patients, and there were a few, but they were very few and far between. And there was not a single mutation that was found in each of the melanoma patients. And bearing in mind that this is only melanoma, and if you move to lung cancer and colon cancer, the mutational landscape could also look very different between different cancers. So this is a bit of a challenge, you could say, in developing a universal neoantigen vaccine. And it's clear that sort of a traditional vaccine development approach where you have one vaccine that is relevant for all patients, that's probably not going to be the most feasible way to continue in this space. So what we think the next step is to tailor the treatment more specifically to each patient. We're already seeing these precision medicine approaches being used today to treat cancer patients, where the doctors are able to measure various biomarkers on the tumors of patients and thereby direct patients to one or more different treatments. So with the Pioneer model, we're taking this to the very extreme, you could say, and we're basically designing for each patient a specific cancer vaccine that will fit his tumor specifically, his or her tumor specifically, and their immune system, of course, specifically. As Michael very nicely introduced, there also can be differences in how our immune systems react to vaccines. So Andreas showed you a bit earlier about how we built Pioneer using these AI immunology building blocks. And I'd like to just dig a bit deeper and show you how we actually came to deciding that these were the building blocks that were needed for the Pioneer model. So we designed Pioneer to model the mechanisms that go on inside cancer cells that would lead to the generation of effective neoantigens for a cancer vaccine. So the first step is, of course, to identify the mutation within the DNA of the cancer. That is sort of the whole basis for this working. Next, we look to see which of these mutations are within a gene and expressed in the tumor, which of these mutations then actually generate a neoantigen, which of the neoantigens are then presented on MHC. Again, as Michael went through earlier, this is a critical step in order for a neoantigen to be effective and to be able to interact with the immune system. And here we, of course, use our Evaxion MHC building block that Michael very nicely went through in the previous session. Next, we have some methods for predicting which of the presented neoantigens are likely to elicit a T-cell response. And finally, up until now, the first 5 steps, we have sort of been looking at a single cell, but now we zoom out, looking at the tumor as a whole. And it's a known fact that tumors can be highly diverse in which mutations they have in their genomes. This is due to the way they evolve inside the body of the patients. And here we also have a building block that allows us to look at the clonality of the neoantigens and select those that are present in all tumor cells, basically. And finally, doing all this, we get our ranked list of neoantigens, and then we use our vaccine design building blocks to design the personalized vaccine for each patient. So right now, Pioneer is, of course, just one part of a much larger process that goes into creating these personalized cancer vaccines for patients. So here, for example, when a patient is enrolled in one of our trials, the first thing that happens is that we take a tumor sample and a normal sample from the patient. These samples are then DNA and RNA sequenced, and this sequencing data is then the input that we use in Pioneer to design our personalized neoantigen vaccine. The design is then sent forth, and we then have a personalized vaccine manufacturing pipeline that produces the actual vaccine, and then it is then given back to the patient. And in our trials, all our patients receive the personalized cancer vaccine in combination with a checkpoint inhibitor, the current state-of-the-art therapy for these patients. So the Pioneer model is the model that we have used to generate several of our oncology product candidates, namely EVX-01, EVX-02, and EVX-03. And in the following slides, I will show you a bit of data from EVX-01, which is our product candidate that is furthest along in clinical development. So it is currently in phase 2, being tested in a phase 2 clinical trial. But I will show you some data from the phase 1 clinical trial, which, as Andreas said, was performed here in Herlev Hospital. So the EVX-01 is a peptide-based neoantigen vaccine, and it is mixed with an adjuvant, so a molecule that stimulates the immune system, called CAF09b. In this trial, we only enrolled metastatic melanoma patients, and the primary endpoint, since it was a phase 1 trial, was to see that it was safe for the patients to receive the treatment, but also to see that it was feasible for us to create a personalized vaccine for each patient. The patients received 6 shots, and as I said earlier, they got it in combination with checkpoint inhibitor therapy. So we had some really nice clinical data from this phase 1 trial. First of all, we met the primary endpoint, which was the safety and tolerability. So I don't think any patients had any severe reactions to the neoantigens, so which we were very happy to see. But we also looked into the immune responses that were generated in the patients, and we're very happy to see that we were able to measure neoantigen-specific immune responses in all of the 12 patients. When we look at the clinical data, we saw that 8 out of the 12 patients responded to the treatment, and 2 actually had a complete response. So they were basically tumor-free at one point in the study. So we digging a bit deeper into this data, it was great, but we would also like to see if we could distinguish between the patients that did well and those that didn't. And here we thought that maybe the score that Pioneer assigns each neoantigen that goes into the vaccine would be predictive of how the neoantigen would perform in a vaccine. So the first thing we did was we saw if the Pioneer score correlated with the likelihood of each neoantigen being immunogenic, so triggering an immune response in the patient. And we saw a pretty nice correlation with the neoantigen scoring higher being more likely to generate an immune response. Furthermore, we then looked into how the neoantigen scores of the vaccines that each patient received, how that correlated with their response. And once again, we saw that the patients that received the vaccines, which had, you could say, higher quality neoantigens, so higher Pioneer scores, they were also much more likely to get a benefit from the treatment than those that received the, you could say, the low-scoring neoantigens. So this is all well and good, but we also decided we were going to look at this at a more unbiased way. And here I guess you will recognize this plot from a couple of other presentations. But what we then did was we decided to divide our 12 patients into 2 groups, so kind of unbiased, those with the highest scoring neoantigens and those with the lowest, and then see how did each of them fare. And as we can see, you can appreciate the blue curve, that the 6 patients that had the highest scoring neoantigens, they had significantly longer progression-free survival. We did the same with the tumor mutational burden, which is sort of the traditional biomarker you use within cancer immunotherapy. And at least in our trial, we saw no difference between those with a high tumor mutational burden and those with a low, indicating that Pioneer is really, it does contain some extra information or is maybe specifically relevant for the neoantigen cancer vaccines. So this data set was really what you'd say is the clinical validation of the Pioneer model as well as our AI immunology platform. Now, we've been talking a lot about the Evaxion HC building block and how central it is to our models. So we really wanted to hit that home. So to do this, we designed an experiment where we have our Pioneer model, and instead of using the Evaxion HC building block, we switched that out with 2 other similar prediction tools for predicting MHC presentation. So one, which is our old gold standard prediction tool, which is based on the NetMHC architecture, so very similar in performance to that. And another highly cited prediction tool called MixMHC-Pred. What we saw when we did that was that the precision with which we were able to predict these immunogenic neoepitopes, it dropped. So we were so clearly seeing that Evaxion HC and the newest version of Evaxion HC is critical in order for us to get the optimal performance out of the Pioneer model. Good. So in summary, Pioneer model is our model for designing personalized cancer vaccines based on neoantigens. It's been tested in 2 phase 1 clinical trials and is being tested in an ongoing phase 2 trial. We showed that the Pioneer scores are predictive of how likely a given neoantigen is to be immunogenic, and are also predictive of how likely a patient is to achieve a clinical response to the treatment. And finally, we showed that Evaxion HC is a very key building block in the model. And now I'm ready for questions.